Otherwise, I think we have a lot of really interesting issues that have come up today, even for Wake Up Sheeple 111, I think would be impressed. 1111, okay, would be impressed with what we've talked about today. So, with that, questions.
I'm Amos Korchin from Tel Aviv, and I want to ask whether we don't perhaps pay too much weight on the anatomy. So human beings have changed tremendously over the past, let's say, 50,000 years. So if we compare humans in the Stone Age and today, we're so different in terms of our behavior, our culture, maybe even our intelligence, without any change, I suppose, in the anatomy. So, and here we study the anatomy of whatever, from flies to humans, and we think that we can, from that, derive understanding of the behavior.
I assume that's to me, yes. Is this? Should be on. Okay. Yeah, so, well, I mean, first off, there are a lot of changes that have occurred in that period. For example, there's a lot of dietary sort of physiology changes. There's differences in olfactory receptors. There could well be changes in brain growth stuff. But I take your point, though, that I don't believe that there's been really any fundamental reorganization of the brain at that point. But we don't need too much. We need, maybe some stretch receptors in the abdomen changing where they're attached, or the time course of recovery from trauma extended by maybe what could be a rather simple cellular mechanism. So I'm not requiring anatomical changes, just a different kind of coding. And I also have no speculations that I'd be prepared to defend at this point about just when this occurred, but it had to be after you could ask for help and get some. So.
Thank you.
Hi, Andreas Kalkan from Sweden. My question is actually also about the anatomy, but from a different attitude now. Actually, like when we look at the evolution or like the development of the brain, for example, if you just compare to monkeys, then most people point immediately out, oh, look at the frontal cortex, it's like 10 times bigger, it's like extended. But it's just like, so to say, bigger, it's inflated. But we know also, I think, from other literature that for example, the inferior parietal cortex might contain much new areas which don't have a homologue to the monkey. And it seems that maybe that kind of areas seems to be like also very interesting candidate for like certain abilities we ascribe like to higher human cognitive functions, but usually people don't say a lot about that. What do you think about that?
It certainly is the case that a lot of human functions, that planning for the future, itineraries, that sort of thing, do depend on the integrity of frontal cortex, and it's larger. So there's two things. First off, Harry Jerrison showed back in the 70s that the frontal cortex in humans, like the whole cortex in humans, is exactly the size that it should be. So each particular subpart of cortex has its own allometric line. S1 and A1 have the flattest slope, and the highest ones are posterior parietal and frontal. Now, what happens as cortex gets bigger is that these areas begin to fission off, and we've actually looked at how many areas per volume of cortex, so it's an interesting line. It looks like it goes up kind of linearly to about a ferret-sized brain and then flattens out. So I'm not quite sure exactly whether areas, there's a belief that areas are a particular input-output function with, committed circuitry, and there, I'm sure a few cases of that, MT would be a good case, for example, but committed circuitry and the rest of them, in the sense of, particular transmitters, assembling information over certain sizes that are that you can demonstrate are connected to function. I've been looking at that for a long time, and it's hard to find any real examples of it. What you see is just this proliferation of size and areas.
So you rather don't think that the inferior parietal cortex might be a generally new area?
No, it's not new. So it's a field homology. That whole parietal region
it's just outstretched.
Is like the original parietal one, and then as it gets bigger, what happens to large areas of cortex is they begin to subdivide into areas by a particular function.
Okay, thanks.
Hi, I'm Eric from UCAM. I want to ask a question to Bjorn Brembs. In your paper titled Towards a Scientific Concept of Free Will as a Biological Trait, you go on suggesting at the end, and I'll quote you, there may be a societal value in retaining free will as a valid concept, since encouraging a belief in determinism increases cheating, and you cite something. The implicit question here is, do we need free will to be morally responsible, I guess? Maybe. However, the theory you offer us doesn't seem coherent with the assumption that moral responsibility rely on free will. The main problem that I see is that the variation in behavior you're describing as a necessary condition for our free will are only informed by internal stimulus. I won't go into the details, but you say that the agent receives, so to speak, spontaneous plans of actions, that is the free part, and then evaluates these plans with the help of informations he receives from the environment, that is the will part. As much as this sounds exciting, moral responsibility doesn't rely really on the free part of action, but more on the will part of action. So why do we need this free, intrinsic, and spontaneous ability to be morally responsible?
Okay, there's a whole bunch of things there, and I probably would need half a day to address each and every one of them. The first part concerned an empirical finding, and so that's and one of the authors, I forgot the name. One of the authors was actually here. Was an empirical finding that if you instill disbelief or doubt about whatever the individual, the candidates perceived as free will, then indeed cheating increased. So that was a citation to an empirical finding. That, of course, has to be seen as completely separate from any considerations of what we would like to call free will. This whole paragraph in the paper that you were citing was basically about whether it makes sense to develop a scientific concept of what people used to call free will as a dualistic concept, right? Because dualism has been dead for a good couple of decades, and so one might actually think that it would be worth ditching it and using something else, a different kind of term for the possibility of choosing between alternatives. So that's a shorthand to the first 2 points, and there was a third point in there about internal stimuli. This is something that I've always that come across very often, and I've always wondered what is an internal stimulus. So, for instance, we can take the brains of all kinds of mollusks out of there, and it works with flies as well. It's just a little bit more tricky. We can take them out and put them in a dish, and they still produce spontaneous behavior. So that would mean then, if this is still internal stimuli, this means that the brain is generating its own. So the nervous system is generating its own stimuli, and then it doesn't really make any sense of talking about stimulus, because then stimulus is anything. And so I really don't see what someone could possibly mean by an internal stimulus, because that's exactly what brains are doing. They generate variability by themselves internally. And so I think that's exactly what we're trying to find out, how it works. How do brains do that?
But my question was how that is relevant to moral responsibility.
It is not related to moral responsibility as the, it's indirectly related to moral responsibility. Moral responsibility is something that comes on top of the possibility to act. So if an action is determined entirely by, or to a large part, by components outside of the individual, then you would call it a response. And you cannot, it's hard to come up with a rational idea about responsibility when you have a causal chain leading up to that response. However, if you can assign the cause to the individual, a rational concept of responsibility becomes much easier. So it's a necessary but not a sufficient criterion for responsibility. So if we wouldn't have the ability to act, then there would not be there wouldn't be a discussion about responsibility. Since we have the possibility to act, we have a necessary but not a sufficient criterion for responsibility. So then a lot of things come on top of that to come to a rational notion of responsibility, but it's by far not everything. But without it, we wouldn't need a discussion.
Jennifer Mather, University of Lethbridge. My question is also for Bjorn and also about free will. But I wanted you to get really much more basic. So I'll try 3 questions and let's see how far you go. What is free will? Where did it come from evolutionarily? And what the heck has it got to do with consciousness?
So the first question was what it is. What is it? Well, I've tried to define it biologically, and from a neurobiological perspective, it's the possibility to generate behavioral options. And then how did it arise, and what was its purpose evolutionarily? I think it arose from behavioral control problems. Initially, probably the primeval reason, or adaptive reason for movement has been dispersion. And as competition increased the pressure on controlled dispersion, there was the need to develop processes that can guide the movement. And one of the first processes that have evolved, I've linked that paper somewhere on the blog, I think, has been Phototaxis. And so this is where one can see the first. What happens there is that these animals, in the paper that I linked to, they first behave and move around randomly, and light comes on, a light source at a particular place, and then they all swim towards the light source. And this is what these colleagues describe. This is done by the light inhibiting certain components of the movement apparatus rather than triggering anything. And so basically you have a set of movement options, in this case realized by moving variably in space, and then this spectrum of behaviors gets limited by sensory stimuli. And this is also something that has been retained. This is what one can see in a lot of animals, where you can have highly controlled behavioral situations. If you increase the number of stimuli, you decrease the behavioral variability. And what that has to do with consciousness, again, I think the relation to consciousness is very indirect. I should preface that by, again, emphasizing that I don't really know what consciousness is, and a lot of people with whom I've talked over this past week disagree with my idea of what I think it is. But if we wouldn't have this option, so it goes a little bit towards the question of responsibility. If we didn't have these alternative options of action, we probably, and there, I'm really way out there, we probably wouldn't be discussing questions about consciousness, because things would be a lot more mechanistic, would be a lot less flexible, and we probably wouldn't have the need or the possibility or the capability of consciousness. I think that boils down to if we can even agree on what we want to call the capability of choosing among behavioral options in humans, right? So I already got a lot of heat for saying, well, let's not ditch free will just yet. Let's just use it scientifically and not metaphysically. And so I wouldn't presume to say that, okay, now I've said that we can use free will in humans. Now let's use it in animals as well. I'd like to see if I can get any resonance for keeping the term free will and using scientifically. I'm not married to the term. I can use a different term. If someone has a good idea of what to call a non-dualistic capability of choosing among behavioral options, and it's as catchy and as easy to explain to the general population as free will, I'm ready.
Pauline, Université de Montréal. This is a question for the whole panel. A quite general question popped up in my head when Professor Singer presented briefly the two hypotheses commonly found in neuroscience about neurobiological structures underlying consciousness, mainly that consciousness might be either produced and controlled in some specific parts of the brain or in the entire brain. But isn't it a mistake to see consciousness as a thing that is produced and controlled by specific structures, rather than seeing consciousness as a general neural state that is the consequence of a combination of all the other cognitive abilities that do have their own underlying structures and that interact together, and which would mean that consciousness does not really have a proper neural circuitry.
Oh my God. Okay. I think I'm going to try to answer that question. I think it doesn't have a proper neural circuitry. I agree. I mean, if you're looking for neural correlates of consciousness and you're using color spots, perhaps you're going to find it somewhere else in the brain. And if you're using random movements or random dots or something like that, I would say one of the things that I wanted to tell at the end of my talk was that there is no such a thing as a brain structure, the way that I see, that every time that you do it and you do an experiment, it's going to light up and say, okay, this is a neural correlate of consciousness. Perhaps you find a structure, but when you go and dig into the neurons that are activated within that structure, you're going to find a totally different set of neurons. Like, for example, in prefrontal cortex, we see that. We find different sets or something that is on the right or on the left or at the bottom and things like that because there are different things. So, yeah, I don't think that is going to be something that is going to be variable. And this is part of the constant. This is part of the problem. And if you translated that across a species, if you're trying to look for consciousness in a mouse or you're trying to look for consciousness in a monkey or neural correlate, and in a human, it may get even worse because you may not even activate the same brain areas or the homolog areas in different places. So that said, it is possible that consciousness has different levels of complexity, and what you would call consciousness in a human may not be the same as in a monkey, or has not been the same as in a mouse or in a fly, or you can go down to as much as you want. So it depends how do you define it. So as a neurophysiologist, the way that I say is, yes, it doesn't have a specific neural circuitry. Maybe there is a structure that could be across a species that play a role, but play a role in the activation and in the whole process that has to do with it. And if you activate this structure alone, probably you won't get consciousness either. So you have to activate the whole structure, and it's going to change from experiment to experiment.
Okay,
That's
so you would say that consciousness is not really a thing itself. It's not something that is produced by a specific system. It's more something that is the result of the combination of several other abilities.
Yeah, I would say the way that I see is the result of the combination of the activation of different part of a system that is highly distributed. And it does, even if the result is perception, you can say, well, I call it perception of motion, perception of color, perception of, and you can keep going with that conscious perception of. And the only thing that is common in those things is the word perception. Because when you look at brains activated during motion perception or during color perception, there are different areas. And probably different neurons within an area. Even if you find an area that is activated across them, you find different neurons. So that's, yeah.
I'm still confused about, when I talk about a neural correlate of something, I imagine one axis is my neural activity or my populations of activity or my different neural areas. You can put anything you want on one axis. But the other axis, you put what you're studying. So we want to put consciousness on that axis and move it back and forth and then see how our other neural correlates move together, and that's how we find these correlations. And that works for things like wakefulness. You can move sleep, wake, and I can have neural activity go up and down, or change from these different regions in the reticular activating system and other places. Okay, and I can see these correlations, or I can have visual perception moving back and forth on one axis and neural activity moving on the other axis. But no one's told me what to move back and forth to study consciousness. Okay, I think it's things like visual perception, but I've never had anyone say, oh, for consciousness we need to measure this, and this back and forth, and then we'll see which areas in the brain also move along with it, and we can form our correlations. So that's my stopping area from the very beginning. The part that I find difficult to start off with is when we want to call a neural correlate of consciousness, what are we varying to measure these correlates? Okay, visual perception is a good one. I think that's a good one, and maybe some of you agree, but there's probably a lot of other ways you could do it that might be even better. So.
But isn't it a mistake to only study one specific structure in the brain?
Oh, no.
Instead of seeing consciousness as a whole, and you can't study consciousness if you don't study the whole at the same time.
I think it's wide open. Too bad Wolfsinger's not here. You can vary perception, for example, visual perception on one axis, and then vary the resonance or the synchrony between various areas on your other axis. I mean, you can put whatever you want to define on your y-axis, but there's plenty of things to put there. Different areas, how they interact, single areas, but it's the x-axis. I don't know what to put on that axis when you say consciousness, but if you say visual perception or wakefulness or certain motor planning or movement, then we know how to measure these correlations.
If I could, I've got one. Channel Bjorn Merker very poorly for a moment. I believe he would argue that we need to look first for the set of probably thalamic mesencephalic structures that generate a egocentric map that has both perceptual, motor, and motivational concepts to it, and we haven't thought about that structure in that way. And then I think a lot of the other things are going to be things that populate that representation with its content. So I think I'm going to go for a much more physical account of what to look for than these others, I think. So. Yeah.
Well, I mean, I guess I'd be more on the side of what Eric was saying. In fact, I heard what I thought was a good analogy, which is that consciousness is kind of like the sky. So if you say that you're interested in the sky, almost everyone knows what the sky is. It's sort of a very nice term. It conjures images to mind, but you wouldn't study the sky scientifically. You wouldn't necessarily have a conference about the sky. You would have a conference about the physics of light or the climate aspects of refracting light or the cosmology of things that produce light or the visual perception of color. So if you just sort of ask yourself, what is the sky, you end up with every person who's involved in the discussion having their own idea about what it is, and every one of those ideas is right, because it is all of those things, and it's the interaction of all those things. So your point about the conscious, well, everyone over there, I'm not disagreeing with you, but the point about the fact that it could be distributed across brain regions and the fact that it could be different for different types of paradigms almost has to be true until you have a very much more precise kind of definition of what you're talking about.
Right over here. Hi, Josh Sanchez from University of Wisconsin-Madison. This question is mostly for Eric, Julio, and Bjorn. So this idea has been thrown around a bit about how when you're doing animal studies, you're having them do a task that isn't something that they would encounter in the wild. So then they're forced to make an association that they wouldn't naturally make. So this has been brought up And I don't really have a background in LFPs or animal studies, but I was wondering if it would be possible or interesting, well, to back up just a little bit, it seems like you train your animals for a task and then you take recordings of like how this new network that you've created works, or this new association works. And I'm wondering about the potential to somehow measure during that process of learning to see if you can see the association being created in the neural networks, or see if like this new circuit will pop out during that training process.
Yeah, I think that is obviously this is a great idea. I mean, learning is another field that people do a lot of research on in our area. One of the difficult things is when you do long-term learning, you have to test on the same animal for a long period of times, and you have to keep probably the same neurons, the same network. So you have to monitor the same network and see how the weights change for a long period of time. We got to the point where we can keep a good set of neurons during, let's say, 3-4 hours when the animal is learning something. So that's the stage where we are now, and we're doing some experiments on that, and that's the. But has been a difficulty in the field. So because you want to do recordings in awake animals, and at the same time you want to isolate single neurons, because LFPs are not that informative about what the single neurons are doing. So it's a difficulty to assess long-term learning. When you're looking at working memory, that what you saw the data, or short-term learning and things like that, is easier to do. And even if you record from a chronically implanted set of electrodes, and you record today and tomorrow, it's very likely that you're recording from different neurons. So that is the difficulty of keeping the animal alive, being not invasive when you do your recordings and your things, because we also have to make sure that the animal is healthy and all that kind of things that go with the protocol for research, and it's very difficult. So I think that you will see a lot of things coming in short-term learning, probably over with the chronic implants that now people keep for several hours, even a couple of days or something like that, over the next, I think, years. Long-term, I'm not sure in the monkeys. I mean, it's going to be a tough one.
I can answer very quickly for invertebrates. Yes, on basically all accounts, we can both monitor and interfere with virtually all neurons involved in learning and memory on various timescales.
Go ahead.
Hi, I'm Juliette, University of Montreal. My question is for Dr. Finlay. I was wondering, what do you think about the idea that the actual anatomical ability for speech would be a distinguishing feature in the evolution of cognitive abilities in humans?
Well, I mean, it definitely is a distinguishing feature. The distinguishing feature? What is the sense of your question?
Well, when we look for what allowed the evolution of cognitive abilities, that would be like a jump that allows them to do.
Both in birds and I think in humans, there's more distribution of telencephalon directly to hypoglossal and some spinal motor nuclei, possibly more than you'd get by having a larger cortex alone. So there could be some change in axon pathways, maybe some speed.
Well, I was thinking about it.
So I guess what's the?
I was thinking about it in terms of the fact that you mentioned a lot the role of social interaction, for example, in pain, and so to me, I thought speech could be related to social interaction, and therefore to a lot of evolution of cognitive abilities, perhaps consciousness as well.
Oh, I see. Well, one thing that's interesting about getting a full representation of the viscerosensory and gustatory system to the cortex is perhaps then you can talk about it. So you're getting this basically the way I see that in a mechanistic term is you're delivering that sensory array to the big autocorrelator, and which is the same structure that is going to be finding categories in language. So I can see how these two things could conceivably interact.
But not necessarily be causal or have a main role in.
I can't hear what you said.
Not necessarily having a main role in the evolution of human
Well, it's
abilities.
just a question of, do we want one role? I mean, I think there's a whole lot of.
Yeah, but I'm thinking of it in terms of contrasting because we look for neural correlates. So, but then, as I understood, you said it doesn't seem like there is so much difference in the brain itself. So, could it be that the difference could be actually something else, like the ability for speech, which once you can speak, then
change, right?
Sorry,
that has to be a neural change, right?
Well, as I understood it, you can teach sign language to chimps, right? But they cannot speak. So I was thinking that this should have an influence on how the infant, how the infant develops. The infant doesn't grow up with a complex language, whereas the human infant does.
I think you're perfectly right. I actually wrote a paper about this kind of point a while back, which is I tried to find every primary cause ever published about what's special about humans. Okay, and you come up with a list of 20 or 30 things in different classes, which are going to be language or social interaction, planning for the future, tool use. I get a proposal on one of those in BBS every week. Okay, and I'm not saying that to minimize your point. It's just that this, I think they're all true. They're all important components. So you can either, I think, try to direct yourself to what is an unanswerable question of which is the very first, or find out what coordinates them. And I'm going through the whole brain structure.
It's just, I thought you meant in your talk that, I don't know if I understood correctly, but I thought you meant that it's very difficult. It seems like there isn't really something special about the brain itself, right?
Well, I saw that in general, in the size, there is not, but there is something really special reorganized in primates. I don't know about in humans. There's no comparative studies of monkeys versus humans in organization of the insula. But I told you about 3 separate ways that the organization of the insula differs in primates compared to rodents. So my point was that brain size were very predictable. Organization of this pathway seems unique. So I was not trying to make it argument that there are no differences in brains. You just have to look for the ones that are informative about niche-related changes, and that's not usually whole brain size.
I see. Thank you.
On the left. I am Felix from University of Montreal, and my question is for Professor Eric Cook. And I was, first of all, you showed a feed-forward model, but you didn't show any feedback model. Is that correct? All right. Okay. Yeah. And I was wondering also, what was the aim of doing such a model? Did you aim at proving that the feed-forward processes are a sufficient condition for causing the phenomenon you were talking about, or were you rather talking about necessary conditions for such phenomenon? So I asked the question because I'm wondering why did you do a feed-forward model instead of a feedback model, and in either cases, whether you try to prove that it is sufficient or necessary, I don't see reasons for doing a feedward model only, and not both of them.
It comes down to something much more mundane. Is there's an infinite class of models that would account for our data, and so I think it's the most informative model is the simplest model. Okay, that can explain your data, and so. I certainly you could add in feedback mechanisms from attention and or changes in internal state, and you could still account for our measurements, but I don't need to. okay, at this point. Now, that does not mean these kind of signals are not there when the animal perceives the stimulus from when he doesn't perceive the stimulus. And at the very end, I tried to add in a little, looking at some of these different frequency bands of LFPs, where maybe you can start to think about these feedback signals that arise from the changes in internal state, which may be related to things like perception. But just to explain the link between the neural fluctuations in behavior, the feed-forward accounted for most all of that relationship. So why go any further?
Why would it what?
Why add more complexity? Feedback would be more complexity. Why add more parameters?
Yeah, I understand.
It comes down just to that.
Yeah, but there's some evidences of feedback processes also. So ultimately, any model of cognitive processing should, I think, to some, maybe not in the experiment you did, maybe in that kind of processing, feed forward are sufficient. But ultimately, to explain maybe other processes, feedback model would be feedback. Yeah.
Right. So we put the animals in a very stereotypical task, where they were looking for a signal buried in noise, and there was, they put their internal state, which is where maybe all the interesting things are happening, into a certain mode of doing this, and this is where we made our measurements. So what you are proposing is, let's have a more interesting task, or something, again, on this, let's vary something that's closer to what we might think about what consciousness is, vary a task, vary some kind of visual perceptual experiment, and then set ourselves up where feedback signals, changes in internal state become critical.
Mm-hmm. All right. Thank you.
Hi, Xavier Derry, Université de Montréal.
I want to add something to you, what Eric. I want to say that not all processes require feedback. For example, if you're talking about reflexive saccades, you talk about just very simple acts, you can just have a feed-forward model and explain it. So it's not always required.
Good. Sorry. My question is for Professor Finlay. From my understanding, the more complex and multivariable a system becomes, the more chances it has to go awry, to go out of control. Maybe not.
Do we know that?
Well, it's my impression. I mean, I've never heard of a schizophrenic aplesia, for instance.
Because you don't know them. Yes.
Well, maybe. But actually, if the answer is yes to that question, it might actually reinforce your point. So your theory about the evolution of the specific pain system for humans as having the function of enticing others to help us, therefore increasing individual fitness. Would you think that chronic, debilitating pain that doesn't seem to have any reason or doesn't add anything more to the fitness of the individual, but is very detrimental at the opposite, do you think it would be an example of a little part of the variation, that extreme where the system that has evolved in some form just goes terribly wrong and out of hand?
Yeah, I've spent a while in the last couple years looking through the medical literature on just that kind of thing, and it hasn't really been considered. In fact, the whole placebo literature isn't considered a great deal in terms of mechanism. And I think if you put this different frame around a lot of diseases, it may be easier to understand a whole lot of syndromes as sort of disorders of the help-seeking mechanism, as opposed to the disorder of a pharmacological component of GABA or some particular part of the pain circuitry. So yeah, absolutely, but this hasn't been done yet to think of things.
Right, it's kind of hard to measure.
Yeah, I think it would be really, that's exactly what I would like to encourage people to think about, because that's not something I know much about.
Thank you.
I'd have a wild speculation in that direction. If you were able to closely monitor 7 billion individuals of almost any species, you'd be surprised what kind of disorders you would be able to find.
I have just a question for Barbara Finlay also. You're very popular today, it seems. It's just really a trying to pick your brains in the hopes of finding something that explains the difference in human and primate brains. Steve Weiss has made a big deal about the granular prefrontal cortex, suggesting that it exists in primates but not in other mammals. Is that actually, from your knowledge of the literature, which is much greater than mine, is that, do you think that's on the right track, or do you think that's another case of a predictable size-like relationship that doesn't actually have any functional implications?
Did you say agranular or
granular?
Granular. Well, the insula thing I just showed you is an example of granular, not frontal, but sort of in the general region cortex that hadn't been there before.
Well, he's pointing to a large percentage of the prefrontal cortex there.
I'm not sure what the case is there, because I never became interested in this granularity issue until very recently. But Mike Shadlin the other day was arguing that the change in brains was exactly the opposite of that, which was really my belief to begin with, that what was expanded the most was the agranular part. So I guess I don't know what the data really are. Sorry.
Hi, Vincent from UQAM. So my question goes to each of you people who use the attended versus unattended stimuli. I asked this question at the panel 2 days ago, but couldn't get an answer. I'm wondering if one of you could possibly answer this. So we see a clear difference between attended stimuli and unattended stimuli. Now, we have a good idea of what attention is, but do we have any idea of how it is created, or how it is, what are the processes that explain attention? Because to me it seems as esoteric as consciousness is right now. I mean, he's attending the stimulus, so well, that's why we get this kind of response. But I mean, what explains attending a stimulus? I don't.
That was personal.
Actually, no. Actually, it wasn't. Actually, it wasn't at all.
I had this discussion also with Paul Sisek, actually, and I got disappointed when he told me, attention, that doesn't exist at some point. I said, well, maybe we're calling the same thing different names. So the way that I say it is a process, is a brain mechanism. The problem is that when we conceptualize things in psychology and we put it in boxes, and then now we want to bring it to physiology, and now we have a problem that the boxes, they don't fit in very well, or the boxes overlap a lot. So then physiology is a very hard thing when we have the dialogue with psychologists or clinical psychologists, sometimes MDs too, because they categorize diseases. So attention, the way that I see is as a filtering mechanism. So the brain has a limited processing capacity, and you need to filter out information. If you don't filter out information, you end up with ADHD or with some kind of, for example, autistic kids, that they can filter out things, and then your performance in normal tasks is not going to be good, or it's not going to be. That said, a child with an ADHD who's just going to do just good being a hunter probably is going to be a good one, because he killed an animal and got to kill the other one, got to kill the other, got to kill. So it may be something that is evolutionary, and we call it a disease, of course, or we call it a brain condition and things like that. So attention, basically, I see as a filtering mechanism. There are two forms of attention. One is exogenous, and the other is endogenous. The exogenous is the one that you call a pop-out stimulus. So if you are here and someone shouts loud there, your attention goes right away there, because there is something important. What that means is that you're favoring just a stream of processing from that retinotopic location into your brain, and the other things don't receive the same priority of processing. That's the way that I see. The way that you can implement that is by having control areas, for example, in the frontal cortex, where you hold memory representation, or working memory representations, exogenous attention is a little bit different. You can implement it through lateral inhibition and through things like that, winner-take-all mechanism in an area. Voluntary attention is a little bit more tricky. So you need control areas, and you need to know what you're attending to, put a mental representation of that in working memory, and then you can route the information from the locations that you want, or the features that you want, or if there are color areas, you can just bust the gain of neurons so that when the sensory input comes, for example, the neurons are going to be more responsive and going to encode the stimulus better. That's the way that I see attention. It's a mechanism and has different flavors. One is endogenous and exogenous. And then we keep splitting it. Spatial attention, feature-based attention, object-based attention. What does that mean? It means that the mechanism perhaps targets different parts of the brain, different group of neurons, and that's how we call them. But that's how I see attention. It's a filtering mechanism, and it also has an amplifier added, which is what I was telling you about the contrast.
Well, that all makes sense, except we can't that doesn't explain, I mean, I understand how it works, but how it is implemented? I mean, how come does one specific stimulus grabs my attention, another doesn't? And how do I decide? I mean, is there free will in there? Or some kind of other force that says, oh, now you put your attention on this?
In exogenous attention, there is very little free will. You can help. If someone shouts your name or something like that, it's going to go there. So if that is what you're referring to, if something very bright happens, your attention is going to go there. I mean, you can do experiments on that, try to block it, try to do things, but there is little that you can do. In endogenous or voluntary attention, you can directly where you want it. And where you want it, I don't want to go back because I'm going to go into free will. I pass the ball, don't want to do that, but you can direct attention where you want it. Let's say if you're looking for, you want to eat, you're looking for some sign that means food, second cup, something like that. So, and that's where your attention goes, and you're better at detecting signs like second cup look like. You're going to be very good at doing that, and very bad at doing everything else, or things that are very different from that.
You say you want food. My question is, who is you? That's actually my question.
Oh, well, me is my hypothalamus and my glucose levels that went down, something like that. That's the best answer that I can give you. Now, my frontal cortex knows what to look for, and which neurons in visual cortex you should enhance the processing so that you will detect those signs. Who is me? Well, I know who I am, but I pass that to Bjorn if he wants to talk about free will. But attention is implemented through connections. I think very likely through feedback connections, the voluntary attention, from frontal cortex or parietal cortex toward visual areas or sensory areas in general.
Okay. Well, thank you. Sorry.
In fact, there are similarities, and I think Paul Chisek pointed that out yesterday, between selection on the sensory side and selection on the action side. And those go, well, depending on your expectations, surprisingly far. So certain dynamics in selective visual attention in flies, if you can record those by local field potentials and match them to, if you wanted to, behavioral output. And what you find are dynamics that are reminiscent of each other. So there's no direct evidence. But it's not inconceivable that indeed conceptually similar selection processes are at hand when selecting actions and when selecting objects out of the environment by using attentional or attention-like processes.
Okay, thanks to you both.
Okay, we have time for a couple more. Go ahead.
Hi, the question goes to Chris Beck. As often when you develop models of the neural system, you can find some solutions in which you have a model that explains the data, but you don't find the implementation of the model in the brain. And this is pretty much what you seem to. You found a model that can explain some of the data from V1 to up to MST, but if you don't find any neurons that implements the nonlinearity the almost binary nonlinearity that you have found, what does it mean for the validity of your model, just from a scientific point of view?
Oh yeah. Well, I wouldn't say it. So this is a very general philosophical question about whether you can map the elements of a particular theory onto actual things that exist in the world. So in a sense, you might say we don't care as long as we have a model that fits the data, but that would be very silly because we're also experimentalists and we want to be able to go and find the elements and get a better understanding of them. So we've hedged against that by using as inputs to each model the outputs of neurons from which we have decades of experience in studying them, these especially area MT. Now, when the model doesn't work, we threw in this set of hacks that make it work, but the hacks are I don't think it's really fair to say that they don't exist in the brain. In fact, they're found constantly in the brain. These are just nonlinear operations that one can find everywhere. What we can say is that you can observe them with extracellular recordings from single neurons in, say, MT. If you wanted to make that claim, you would find neurons that had bizarrely non-physiological tuning curves. So, to me, that's a really excellent use of modeling, because now I can go in and look for this as an experimentalist and see where does this occur, and I've narrowed it down quite a bit. Either I have a completely wrong model of MT, which would be kind of sad because this is like the most thoroughly studied area of the brain, or there's something that I'm missing which happens, and I can say that it has to happen between the spiking output of one neuron in, say, MT and the input to an area like MST. So to me, this makes a very strong prediction for something like synaptic depression, which we can now, and in fact we are now trying to figure out how to study. So that could turn out to be wrong. Our first intuitions about the system were wrong, but if it's right, then I think we've learned something through the modeling, and that's what I think ultimately the modeling, that's the most you can ever hope for.
Okay, that's good. Thank you. Diego Mendoza from McGill University. So a couple of the speakers referred often to the neural correlates of conscious experience. And this is a term I think is inherited a lot from the legacy of Crick and Koch, who back in the 90s very gently and very shyly proposed that you could actually go in the brain and find that. Now I think the word correlate was very well thought by them, not to create too much of a response, just to say, okay, the first thing that scientists look for is correlation. I think we should stop that shyness and just go and say what we're looking for is the neural causes of consciousness. And that, I think we're getting, I mean, there's a lot to be done, but we're starting to get ready to do that by, developing techniques of causality, like, lesioning studies that can be down to the cellular level, up to brain areas, using now the newly developed optogenetics and other things to actually inhibit or manipulate the system and then cause normal function, normal conscious function to either disappear or reappear at will. So I'd like to know what you think about that, and how we can actually, maybe by manipulating the brain with these things, not go beyond just causality and say, okay, these particular element, a neural element or a neural process is necessary, and a particular set of neural elements are not just necessary but sufficient to support a particular conscious experience.
Well, I guess maybe I don't understand how this is different from the standard microstimulation experiment. So the microstimulation experiments prove that the animal sees something and reports on it. So operationally, I'd say that's pretty strong evidence. Now, are you sort of weaseling about what it means to be conscious of what the animal is seeing, or what else would make you happy?
No, I'm just talking about, no one talks about the neural causes of consciousness. Everyone talks about the neural correlates of consciousness. I think that's very, very diplomatic, but I think we can be strong enough to say, okay, we look for the neural causes of memory, of attention, of this, of that. Let's go ahead and call it the neural causes of consciousness, or of a particular conscious experience.
I'm not sure it's an entirely sensible question. I mean, you could go and stimulate the brainstem, the reticular formation. You would find something that caused or abolished consciousness, but I don't know if that would be a very satisfying answer, right?
It's a conscious experience I'm talking about, not just consciousness in general, but let's say a particular experience of seeing your face in this particular place of the visual field, and that. That particular conscious experience must have some neural events that support it. That's a kind of thing I'm talking about.
All you have is the operational report of the animal with whom you can't really communicate about highfalutin philosophical terms. Then how can you distinguish between a sort of mechanical response, i.e. I've stimulated area IT and with exactly the right pattern to create Diego's face, the animal presses the button for Diego, but he's a zombie, he's enslaved by the microstimulation, versus he's experiencing Diego in a conscious sense. How would you ever distinguish between those two? Or are you proposing manipulating something in a human who can tell you specifically about the subjective experiences of your manipulations?
Well, in binocular rivalry, we know that it's really something that we are experiencing. It's not just an automatic thing. I really see it or I don't see it. And then when you look at monkeys, they respond in the same patterns that humans. So, I mean, it's not complete evidence, but it strongly supports that monkeys are probably doing the same thing or perceiving something very similar to humans. So there are ways to ask animals to report their conscious experiences, I think. I don't know.
Go for
Okay.
it.
Well, we've run out of time, unfortunately. It's 5 after. So let's thank our speakers one more time.